Hello friends, in my last video I had an introduction on the transcription of RNA, and now here I will be talking about some terminologies that you need to know about transcription. One is closed complex, and that is this closed complex. This is my DNA strand, and this is the promoter region. And this is the specific sequence that I want to transcribe. Now the thing is that when this RNA polymerase, this is the RNA polymerase that binds to the promoter region, and this is my core enzyme part. I'm just coloring this one. So together this holoenzyme binds on the promoter region, and this sigma factor is actually involved in identifying this promoter region, which is this portion. And then this is my start point. So now this DNA associated with the RNA polymerase, still it is closed because all these hydrogen bonding between the two double helix is still over there. So when the double helix is not open but the promoter region is associated with the RNA polymerase, this kind of association is called a closed complex, and sigma factor identifies this promoter sequence among the other DNA sequences and increases affinity of this RNA polymerase for the promoter. Now what happens in the open complex, which is our next point of discussion? The open complex is the subsequent formation of this closed complex. Now if this is the DNA strand and here is the double helix, and what happens when this this promoter region, when this RNA polymerase is bound? This is my RNA polymerase core portion. Then it has got some intrinsic property, and it dissociates this two double strand at that particular point. Actually it melts the double strand, and so like the replication bubble, here also one kind of bubble is formed. And this is my DNA, this is the double bond, and in this portion the double bond has been this is my hydrogen bond, and in this portion the hydrogen bond has been broken. And so here my RNA enzyme, this is the sigma factor, and this is the core enzyme portion. And here when the double strand has been opened and this hydrogen bonds has been disintegrated, now this kind of complex is called the open complex. So now the this is the this actually after binding this enzyme at the promoter region, the enzyme rolls to the start point where the transcription will start, and here is this portion is the start point, and the RNA polymerase comes to the start point and opens this DNA double helix and the start at the start point. And this structure, this DNA double helix opened at the start point associated with the RNA polymerase, this is called the open complex. And one more thing I should talk about. In my earlier video I told about the abortive initiation, where 2 or 3 nucleotides has been added to the RNA, and then the RNA polymerase falls off from the DNA double helix, and this kind of initiation is unsuccessful for this transcription, and it is called the abortive initiation. But when the RNA polymerase manages to transcribe, suppose this 10 nucleotide long RNA polymerase, then then it is it can be assumed that this RNA transcription process will continue. And now when this has happened, this DNA double helix, this RNA polymerase, and this RNA together, this three thing, this DNA plus RNA and this RNA pol together, this three thing form the ternary complex. This is called the ternary complex. Okay, so as this RNA polymerase moves towards this direction, opening this DNA double helix by melting this double bonds, by melting these hydrogen bonds here, it will move on. And so the RNA, this RNA will increase in size as it getting polymerized with the new nucleotides. And as it moves towards this size, the DNA double helix starts to denature, and the previous portion, this starts to get renatured. So denaturation towards this direction, and renaturation, that means again the association of the two double helix together is in this direction. So as it moves, the RNA gets elongated, and this is the elongation portion. So now what we have discussed about this closed complex, this open complex, this ternary complex, and this denaturation and renaturation that occurs as the RNA polymerase moves from 5' to 3' location. And this is the 3' end. This is the 3' end of the of this RNA, and sorry, this is the 5' end of the RNA, and this is the 3' end. Let me take a new color so that it will be easy for us to understand. This is the 5' end of the RNA, and that is the 3' end, as because this transcription takes place from this 5' to 3' direction. Okay, so until now I guess this much of discussion, you understood everything. And now we will be talking about this elongation portion. This elongation is actually adding up the nucleotide together. This is the RNA, this is the ribonucleotide, where this portion is the base associated, and in this portion the phosphate group is associated. And what is the difference between the DNA nucleotide and the RNA nucleotide is that this at this 2' region, in case of this RNA nucleotide, OH is present, whereas in case of this DNA nucleotide, this at this 2' position, oxygen is not present. That means only hydrogen is present here, and this is where the base is associated, and here in this portion the phosphate group is associated. So this is the DNA nucleotide, and this is the RNA nucleotide. This is called deoxyribonucleic acid, because this oxygen is not present. And in case of RNA, it is ribonucleotide. There is no story about this oxygen, the presence of the oxygen or the absence of oxygen. So this is the difference between the ribonucleotide and deoxyribonucleotide. Now what happens that this ribonucleotide, all this gets connected to each other from this is the 5' end where the phosphate group is added, and it connects the second nucleotide with the 3' position. This is my this is the third carbon, and this is the 5th carbon. In between them a bond is there which is called phospho diester bond, phosphodiester bond. So this nucleotide 1, this nucleotide 1, and this nucleotide 2, they get connected by the phosphodiester bond between, and the bonding occurs in between the 5 prime phosphate group of the second nucleotide and the 3 prime end of the first nucleotide. So in this way, this direction of the nucleotide occurs, because there is another 5 prime end of this first nucleotide. So in this case, the topmost nucleotide, this topmost nucleotide has got the 5 prime end on the top, and the lowermost nucleotide has got a 3 prime end at the bottom. So this is the 5 prime 3 prime directed nucleotide RNA strand, which takes a DNA strand which is in the direction of 3 prime to 5 prime. Okay, and now this transcription rate is another thing, that how many nucleotides are getting added per second. So generally at 30 degrees centigrade temperature, this 30 degree is centigrade temperature is important to mention, because the enzyme activity also depends on the temperature. So at the 30 degree centigrade temperature, 40 nucleotide per second is the rate for this transcription in case of prokaryotes. So now in this DNA strand, as this this RNA polymerase moves, and one more thing I should mention, that after 10 base pair length of the newly transcribed RNA, this sigma factor falls off, and now this whole transcription process is under the function, or under this core enzyme. So it actually now manages the whole transcription process. So now as it moves, so in this portion, the front portion where it is moving, the positive supercoiling, the positive supercoiling is generated, and this positive supercoiling is removed by the enzyme called gyrase. And it actually when this positive supercoiling is increasing, what the gyrase does, it adds negative supercoiling to remove this positive supercoiling from the DNA, and so that this RNA polymerase can move to this direction. And so in this way, this elongation takes place, where a new nucleotide is added, taking this DNA as the template. And I'll be discussing about the termination in my next video, so stay tuned. Thank you.